Thank you very much for your kind invitation. I'm very pleased to be here and to talk about one of our research programs in tertiary lymphoid structure that I co-lead with my colleague Jean-Luc Theillot present here today. So as you know, the tumor microenvironment is a very highly complex milieu and highly heterogeneous regarding different components of the tumors and also regarding the immune infiltrate. And so many years ago, we were very lucky to see that when you look at the immune microenvironment, you can see that in some patients, in some tumors, you can see lymphoid cells that can aggregate together. And here you can see the highest level of organizations that we name tertiary lymphoid structures, TLS, with a T cell area here adjacent to a B cell area. And you can see that all the cells that we used to see in the lymph node, for instance, are present here in TLS in the tumor of some patients. We were also very exciting to see that when you take into consideration the presence of this TLS in lung cancer patients, for instance here, you can see that more you have, you can see that more you have mature TLS with both T and B cell area, better is the outcome of the patient, even in a very advanced stage of the disease here. And so you can see that the median survival was not reached compared to patients having low density of mature TLS. So now this observation has been observed in many other solid tumors, for instance, breast cancer, even triple negative breast cancer, colorectal cancer, renal cell carcinoma, even in glioblastoma. More recently, in melanoma, in CCRCC and sarcoma, and more recently, and as illustrated here in advanced metastatic NSCLC patients, you can see that the density of TLS can also predict the response to immune checkpoint blockade. As illustrated here, you can see that patients having high density of TLS signatures, better response to anti-PD-L1, the survival is better compared to the other group or patient treated with docetaxel. Interestingly, in this publication published by Italiano in Bordeaux, Institut Bergonié, they have shown in sarcoma patients that if you pre-select patients according to the density of TLS, you can have a better response to anti-PD-L1. Indeed, here you have all-comers patients where you can see that unfortunately anti-PD-1 has no clear effect, not enough. But if you pre-select TLS high sarcoma patient, the objective response rate is very better, higher than in these unselected patients. So knowing that TLS is a powerful prognostic and predictive biomarker in cancer, in many types of cancers, our objective is to try to induce TLS in order to improve the survival of the patients. So first, to try to find the way to induce using cells or molecules in patients in order to modify the tumor microenvironment, and then to try to convert cold tumor to hot tumors, and then in combination with ICIs, try to overcome resistance to this strategy. So how to induce TLS? There is some publications in the literature talking about different molecules that have been tested in a preclinical murine model, where you have different members of TNF superfamily. You have many chemokines and cytokines. So indeed, it works in mice, but you have also side effects. and it is not selected specific to TLS. These molecules are known to be involved in other biological functions. Different cells have been positively associated with the presence of TLS, and others, when you deplete, for instance, Tregs, you can also improve, induce, or boost the formation of TLS. In the lab, we were very interested by the innate lymphoid cells, type 3, so ILC3. Why? Because they can do most of the job of these populations. They can uptake antigens, present antigens to CD40 cells. They can secrete cytokines, such as interferon gamma. And they can also kill tumor cells by a direct effect in a trail-dependent manner. So we were very interested by this population. And in addition, when you take into consideration what's happened during the embryogenesis, a subset of IC3 named LTI cells for lymphoid tissue inducer cells, so it's a subset of IC3, they are very important for the induction of the lymphoid organogenesis. So our hypothesis is that in adult, the IC3 may play more or less the same job and can induce the formation of TLS under inflammatory situation. So it's what we are currently testing. So we have set up a mouse model with lung inflammation. You can see that it is very interesting to see that the kinetic of the IC3 recruitment in the inflamed lung parallel the kinetic of lymphoid aggregates and TLS induction in the lung of the mice. So you have exactly the same kinetic. The optimal timing is around day 10 here, and at day 10 you can detect the presence of IC3 within the TLS in the lung of the mice. More importantly, when you sort the different immune cells infiltrating the inflamed lung, and you look by single-cell RNA-seq the expression of many genes, you can see that only ILC3 here express many genes related to lymphoid organogenesis. So we hypothesize that these cells may have the ability to induce TLS in this model. So the perspective is to clearly demonstrate in vivo that the presence of ILC3 is a very critical and a powerful TLS inducer cells. We are testing now the ability of ILC3 also to participate in the TLS-based immunotherapy. And we think that using this strategy, we can induce a long-lasting anti-tumor immune responses following the induction of TLS within the tumor. And the end will be to test this combination with other therapies such as immune checkpoint. So in summary, what we are currently working is to identify different TLS inducers. I have talked about IC3, but we have other projects regarding new molecules that we are testing in vitro and in vivo in order to try to convert TLS low patients into TLS high patients. And the next step will be to combine this TLS inducer with immune checkpoint in order to boost the ongoing anti-tumor immune response taking place within the TLS. So thank you very much for your attention.
Thank you.